Hello and welcome to Bio40 Pre-Lecture 26. Today, we're going to talk about how E. coli is able to utilize lactose as a food source. So why do we care about E. coli metabolizing lactose? Well, we don't really. Metabolism isn't part of this course. But this system is a model for bacterial gene regulation. E. coli, like all organisms, has a lot of genes that are only needed under certain circumstances. Expressing them all the time would be a waste of resources. A gene involved in lactose metabolism only needs to be expressed when lactose is around. So how is the cell able to decrease expression when lactose is absent and increase expression when it's present? François Jacob and Jacques Monod won a Nobel Prize investigating this problem. They studied 3 genes involved in lactose utilization. Together, these genes make up a unit called the lac operon. An operon is a cluster of genes that are all transcribed on a single mRNA. Having multiple genes all involved in the same process on the same mRNA means they can be regulated together. Let's take a look at the lac operon. This is the major portion. Transcription begins at the promoter and proceeds without stopping through 3 different genes, LAC-Z, LAC-Y, and LAC-A, until the terminator sequence is reached. So we get a single mRNA with all 3 genes on it, and ribosomes translate each one. These genes are called structural genes. That doesn't mean they're involved in cell structure. It's just a term that distinguishes them from regulatory genes, which we'll get to in a moment. Additionally, note there's a region called an operator, which we'll also come back to later. Just before the operon, there's a regulatory region. There's another gene here, but with a separate promoter and terminator. This gene, LAC-I, is a regulatory gene. It has no direct role in the utilization of lactose. It simply controls whether or not the cell expresses the structural genes that do have a direct role. There's a key point I want to bring up about mRNA that is synthesized this way. Polycistronic RNA, RNA with multiple genes, has a ribosome binding site in front of each gene. But the ribosome only loads efficiently onto the first site. After reaching the first stop codon, it doesn't dissociate completely, but simply slides to the next start site. That leads to an odd effect, where a nonsense mutation in an early gene prevents the translation of later genes, though this property can be useful in mapping. The conjugation mapping we discussed before isn't very good at distinguishing the order of genes that are very close together. But if a nonsense mutation in LAC-Z stops LAC-Y expression, while a nonsense mutation in LAC-Y doesn't affect LAC-Z, we can conclude LAC-Z is before LAC-Y. The structural genes of the lac operon were discovered by Monod et al. In a process you should be very familiar with. They isolated mutants that couldn't utilize lactose, which we call LAC-, used complementation to determine that all their mutations fell into one of 3 genes, and used mapping to show the genes were tightly clustered. Well, that's not completely true. Actually, they only found 2 genes, which they called LAC-Z and LAC-Y. A third gene, LAC-A, was later discovered, but it's not essential for lactose utilization, and we're actually still not sure what it's there for. But LAC-Z and LAC-Y certainly are important. Mutating either results in a LAC- cell. But such mutants can be rescued with the addition of an F' factor carrying a wild-type copy of the appropriate gene. We now know that LAC-Y encodes lac permease, a protein needed to let lactose pass through E. coli's cell membrane, and LAC-Z codes for beta-galactosidase, which splits lactose into the monosaccharides galactose and glucose. If you're curious about how these sugars are metabolized, you'll really enjoy next semester. Now we know what LAC-A does. It encodes a protein that adds an acetyl group to lactose. We just don't really know why that's relevant. So we'll focus on LAC-Z and LAC-Y. E. coli wants to limit the expression of these lactose utilization genes unless lactose is around. But the presence of lactose should stimulate transcription, followed by protein synthesis. This is called induction, and the small molecule that's responsible is called an inducer. Here is where the extra site we saw before is important. The control site, or operator. Operators can be upstream or downstream of promoters and sometimes overlap. Proteins can bind to these sites and affect whether or not RNA polymerase can bind to the promoter. If the polymerase doesn't bind the promoter, there can be no transcription. And inducers can affect whether or not proteins bind to the operator. The operator of the LAC operon is called LAC-O, and the protein that binds there is called the LAC repressor, which is encoded by the nearby LAC-I gene. So now we have all the key players: the major structural genes and the regulatory gene, the key DNA sites, and the small molecule that signals the need to transcribe the structural genes. Let's look at how the system works. The LAC-I gene is constitutively expressed. That means it is expressed all the time, rather than only under certain conditions. So lactose or no lactose, there's LAC repressor protein in the cell. This protein serves as a negative regulatory element. In the absence of lactose, the repressor binds the operator and prevents the polymerase from binding the promoter and transcribing the structural genes. Which is good, because there's nothing for their gene products to do when lactose isn't present. But when lactose is present, the inducer interacts with the repressor, causing it to change shape and stopping it from binding the operator. That leaves the polymerase free to bind the promoter and transcribe the LAC Z, Y, and A mRNA. This induction leads to a 1,000-fold increase in LAC mRNA and enzymes. Using language like 1,000-fold increase should tell you that the expression prior to induction is not zero. And it's actually important that at least a little protein is produced at all times. If there is absolutely no permease expressed to begin with, how would lactose ever get into the cell to induce expression? Beta-galactosidase also has a key role in induction. While its main job is to convert lactose to galactose and glucose, it sometimes changes lactose to allolactose instead. And this is a true inducer. It's allolactose, and not lactose itself, that binds the repressor and stops it from binding the operator. In labs, we frequently use an artificial molecule called IPTG that mimics allolactose to induce expression. Unlike allolactose, IPTG can't be broken down by the cell, so it has a more sustained effect. Now, if we want to study regulation, we're going to need mutant versions of the regulatory elements. Say we wanted to isolate LAC-I mutants where the repressor protein is no longer functional. What phenotype would you expect from such mutants? I do sometimes miss getting to ask these questions to actual students. Well, if there's no functional repressor, nothing ever blocks the RNA polymerase from transcribing the mRNA, so beta-galactosidase and LAC permease are synthesized regardless of whether or not lactose is present. Expression is constitutive. But it ends up that mutations in LAC-I aren't the only way to get constitutive expression of the LAC genes. The same phenotype can also result from mutating the operator sequence. Changing the base sequence of the operator means the repressor can no longer recognize and bind it. If the repressor can't bind, then once again the polymerase will transcribe the structural genes even in the absence of lactose. The notation for such operator mutations is LAC-O superscript C. So we have two mutants that have the same phenotype. How can we tell them apart? I choose to imagine you responding, "We use complementation to tell us which gene a mutation is in, Professor Brown. Also, you're my favorite professor." Well, imaginary student, you're close, but recall that complementation requires functional gene products. And the operator isn't a gene. It has no gene product. Complementation with LAC-I works as you'd expect. Say we have a strain of E. coli with the LAC-I mutation. It will constitutively express LAC-Z. We can introduce a plasmid that has a wild-type copy of LAC-I. This gene will create functional repressor, which can bind the operator on the chromosome. Expression has been restored to inducible and not constitutive. We call this acting in trans. The gene product on one piece of DNA affects the expression of genes on a different piece of DNA. Here's the same concept from a different point of view. In a haploid LAC-I mutant, only defective repressor is made. It can't bind the operator, so the LAC structural genes are constitutively expressed. But if we add an F' factor that includes a wild-type LAC-I allele, inducible synthesis is restored. The functional protein produced by the gene on the F' factor diffuses through the cell and binds the chromosome's operator, blocking expression in the absence of an inducer. This also tells us that this particular LAC-I mutant is recessive, since complementation would not work if it were dominant. Finally, note that the LAC-Z gene on the F' factor is mutated. That's because we only want to see how the regulatory elements affect expression of the chromosomal LAC operon. Now let's try that same thing with the constitutive operator. If that mutation is on the chromosome, introducing a wild-type operator on a plasmid doesn't change anything. There's still nothing blocking transcription of the chromosomal LAC genes. Expression is still constitutive. The operator only affects the expression of genes that are on the same piece of DNA that it is on. We call this acting in cis. Here's that other view again. In our haploid constitutive operator mutant, the repressor is just fine, but it can't bind the mutated operator sequence. So transcription occurs with or without the inducer. Then we bring in an F' factor that has a wild-type operator. The repressor can bind that operator with no problem, but that doesn't matter since that operator has no influence on the chromosome's LAC operon. There's still nothing binding the operator on the chromosome, so expression is still constitutive. Complementation does not work with cis-acting elements, because they don't produce diffusible products that can influence other pieces of DNA. So such a mutation can be thought of as cis-dominant. So to summarize, trans-acting elements are gene products, like proteins and occasionally RNAs, that can diffuse through the cytoplasm and affect any DNA molecule in the same cell, while cis-acting elements are DNA sites that only affect gene expression on the DNA molecule they are found on. When analyzing partial diploids, trans-acting elements can be dominant or recessive to the wild type, but cis-acting elements are always dominant. So that covers the basics of the LAC repressor system. There are some more advanced concepts I want to get into next time, but first I want to quickly introduce some additional types of regulatory systems. We talked about repressors that bind operators and either prevent polymerase binding or progression, but there are also activators, proteins that bind operators and help the polymerase to bind to the promoter. This means induction can occur in multiple ways. In addition to preventing a repressor from binding an operator, an inducer could help an activator to bind. And there are also small molecules that negatively regulate expression. A co-repressor binds a repressor and helps it bind to the operator, thereby blocking transcription, while inhibitors prevent activators from binding DNA, so there's nothing to help the RNA polymerase bind the promoter. Inducers, co-repressors, and inhibitors are all types of effectors. An effector is any small molecule that binds a protein and affects its activity. So those are the basics. We'll expand on the LAC system next time, and talk about regulatory systems that rely on RNA rather than proteins.